Konnichiwa. Antibodies. These rather famous, important molecules I have been hinting at. Let's take a closer look. Let's pull back the curtain and look to see what is these antibodies. What are they? They're immunoglobulin. So it's globular-shaped protein rather as opposed to linear, like a fiber. And they're involved in immunity, so immunoglobulin. And they're produced against a particular antigen, a specific antigen. And they don't necessarily kill the antigen themselves, but they help out. We'll get to that. They're, like I said, which we've shown already, Y-shaped proteins. They're very soluble. That means that they're very easy to be dissolved in the various humors of the body. They're made up of 4 polypeptide chains, 2 heavy chains, and 2 identical light chains. The 2 identical heavy, 2 identical light. And they just get their name because when they were first being discovered and studied, they would separate them by fractionation, spinning in a centrifuge. And, well, one was heavy and one was light. So, looking at the weather here. So there's a hinge region where there's a little bit of flexibility, kind of like a clothespin. They're held together by disulfide bonds. Remember the disulfide bond? That is formed between the 2 amino acids that are cysteine. Cysteine has as its side chain the sulfhydryl group, and when 2 of them are in close proximity, they can form a disulfide bridge, which is a strong covalent bond. So it holds it together strongly. And there's 2 important functional areas, the variable and constant region. I'm going to think the variable region is a little bit on the more important side. The variable regions are located at the ends of the arms. That's the place where the antigen binding site is. And that's attached to the specific antigenic determinant, whether it was bottle or roundy or pointy or notch. And it forms by weak intermolecular forces, so between molecules. Things like hydrogen bonds, electrostatic interaction is a weaker type of bond, electrical. If I told you about van der Waals forces, you'd think I was crazy. It's really kind of always sounded like science fiction to me. And some hydrophobic interactions, some of the same kind of weak interactions that hold proteins together. Here it is in all of its beautiful glory. So the heavy chain, the 2 heavy chains are held together by disulfide bonds, and the light chains. And we have the variable region with the antigen binding site and another disulfide bond here. So this would be the type of antigen that would be adapted to attack roundy antigenic determinant. And so what this is it's a protein. It's a chain of polypeptides. It's just all wrapped together, polypeptide chains, amino acids, and blah blah blah, which are pretty similar in almost all the regions of the antibody, except in what's called the variable region. So all antibodies that somebody's going to make are going to be, all this part's going to be the same. What the cell needs to be able to do is be able to make what each antibody is going to be, each antibody type is going to be different at the binding site. So this is going to have a polypeptide chain shaped complementary to the antigenic determinant. And then literally what happens is that this will latch onto the antigen, sort of like a little clothespin. It'll open up and the antigen will get in there, and then it'll be bound together. It's really kind of amazing the more you think about it. Antibodies. So they immobilize the antigens, or they cover it up. Ultimately, it's going to be eliminated by other immune cells, the phagocytic cells, neutrophils, and macrophages. they're especially effective in binding to viral particles because, of course, the protein coat on the outside. The cell walls of bacteria and toxic chemicals. Anti. Here's an example of a toxic chemical. You get snake bit, and you get rushed to the hospital, and you get the antivenin. And the antivenin is produced. It's antibodies that are produced to bind to that particular chemical, toxic chemical. Yeast spores. These spores are pretty small too, as far as eukaryotic cells go. And of course, this is humoral immunity. How do they do this? There's three main ways. There's neutralization, agglutination, and precipitation. So I'm going to talk about this slide, and this slide, and this slide from this picture. All right, neutralization. This is particularly helpful for viruses. If we can cover the outside of a virus with antibodies, we're able to perform its function, especially if we can cover its receptor site. In the example of the HIV virus that has a receptor that uses the CD4 CCR5 receptors complex to get into the T cell, if we can cover that part of the virus with an antibody, well, it's not going to work. It's like putting super glue in a lock. Nope, you can't get in here. Neutralized, stopped, right in its tracks. Agglutination. Agglutination is a term that means clumping. You probably heard that term maybe when you were talking about the blood typing test. You were looking for clumping. That was agglutination. And so this is particularly helpful with bacteria because then you can get two of them, a bunch of them linked together, and then call out the phagocytic cells. Hey, come, look what I got for you over here. I got you a bunch of these things. Go ahead and consume them. And the same thing would happen with neutralized viral particles. They would get phagocytized too. So it could be the antibody creates clumping. Precipitation. So for an antigen that's soluble, that's dissolved in whatever fluid you happen to find yourself, if we link them together enough, it might come out as a solid. It might precipitate as a solid, and then that's going to be really noticeable. And agents of the immune system can come over and phagocytize. So whereas the antibody doesn't necessarily, except maybe in this process of neutralization, doesn't kill the antigen, it just gets on it and covers it and then calls for help. Complement. I think we've talked about complement already. Yes, you're looking good today. So it does require direct contact. I've mentioned that. Limited numbers of T and B lymphocytes. Only so many of them are going to be able to recognize the antigen, and then there's this lag time where this all takes time. It takes, first we have to, the naive lymphocyte has to encounter the antigen. Then it's got to find the right receptors that are going to help it make antibodies. It's the right BCR, B cell receptors have to come into play. Then once that happens, once our enemy has been captured and then interrogated, and the B cell then knows how to make antibodies against that antigenic determinant, then it gets activated by a T cell and then proliferates into many, many, mostly plasma cells, some memory cells, but mostly plasma cells. And that's going to take time. And then it can start producing antibodies, which then is also going to take time. But once this gets going, once this whole process gets rolling, and we have all these plasma cells, they make these proteins are not very big, and they can make them really quickly. And one number I saw spit out like 10,000 of them a second. And think how many that's going to produce over the course of 5 days or so. So it takes a while. There's a lag time. It takes maybe 10 days to 2 weeks for this to all happen. But then when it does, it's a very powerful way to eliminate pathogens from the body. Now the memory cells. Let's come back to those. So they retain their knowledge, memory. They remember this specific antigen. And so that the next time we encounter that same antigen, we're already several steps ahead. For instance, a flu season, a regular flu season, not this one. If you go out and you're exposed and you get the flu, what are the chances are that you're going to get exposed to that same flu virus again? Let's say you picked it up at school or at work, and you're out for a couple days, and you go back, it's still going to be there. But you're not going to get sick again, not from that same strain. Why not? Because, well, now you have antibodies against that. And if that same strain gets in your body and starts trying to cause trouble, your immune system says, aha, hold on a minute there. Fool me once, shame on you. Fool me twice, I don't think so. Look at you, Mr. Antigen. See, it's got to be Mr. There's no gender in antigens. All right. Look at you, antigen. I know you, and I've already got I remember how to make, because you have the memory, I remember how to make antibodies against you. And last time it took me almost 2 weeks to get my antibody titer up enough to defeat you, but now I can do this in a day or so. So, you are not making me sick again. No sirree. And that's what this slide says. With subsequent antigen exposure, I think that I said it a little more eloquently, though. Subsequent antigen exposure, the memory cells are already there. They already have their BCRs exposed. This whole thing happens quickly and more powerfully. And I've got a graph that's going to demonstrate this. And you're probably thinking, this sounds like a little bit like vaccinations. And you would be 100% correct. And there it is. It says right there, vaccines typically effective in developing memory. So the way that works is a vaccine typically will have not a whole pathogenic organism. They used to back in the early part, but there's all kinds of problems with that. You don't want to give somebody the disease. But hey, medical science is first exploring vaccines. Things happen. Not all of them good. And so the idea is nowadays what they'll do is they'll typically give you just the antigenic determinant, and it's not in any way the entire pathogen. So there's a difference between the pathogen and the antigenic determinant. The pathogen is the organism that causes illness. The antigenic determinant is part of that pathogen that we can make an antibody against. So they might just give you the antigenic determinant and will cause an immune response, which is why when you get a vaccine, whether it's you, your dog, your kids, they'll tell you, well, they might run a low-grade fever for a couple of days. That's because you're inducing an immune response. And the whole idea of that is to create these memory cells. So here's a rather good graph. There's two graphs. On the x-axis of both of these graphs are days from first exposure to the antigen. And on the y-axis, pretty simple, increasing antibody titer. So let's take a look at the red line for immunoglobulin G. So days from first exposure. So here we are, we're exposed. We've got no antibodies. Still, we have no antibodies. Here we are a week out from our exposure, and we've got no antibodies. So what's taking place during that time is the B lymphocyte, naive B lymphocyte, going on to encounter the antigen, then finding a BCR that fits that antigen, then being activated by a T cell, and then proliferating, dividing into many cells. That's all taking place in this first week here. Then we start to produce the antibodies. Antibody titer goes up, And you can see in the primary response, that's the first exposure. It's still, we go all the way down here, it doesn't peak until 2.5 weeks. And then it goes back down because, well, now we're not sick anymore. We've defeated the invader and we're good to go. And now after this is all done, after the primary response, immunoglobulin levels go back down, we've got those memory cells. And those memory cells remember the whole first week's process already. And there's a bunch of them, not just one. So that on the secondary exposure, the secondary response, look how much faster it goes up. We get exposed, one day, boom, we're starting to make antibodies. And then not only do the antibodies increase faster, but they go to a much higher level and they stay high for a lot longer time. This is the immune system saying, oh, hell no, I don't think so. And why does it stay elevated for so much longer? Is, well, if you got exposed to it once, that's one thing. If you get exposed to it twice, what do you suppose the chances are you're going to get exposed to it a third or a fourth or a fifth time in what effectively is a month here? And as long as you keep getting exposed, the antibody levels will stay higher. So we get a much quicker and a much more rapid response, so that on secondary exposures you might even not even notice. And this is the whole idea behind vaccines, is so that when you get a vaccine, if you get exposed to something, instead of a primary response, you get a first response, and then you might even not know you were exposed. All right, so the initial exposure can be an activation. You get actually real life exposed, or by vaccine. And I've mentioned all this from those graphs, so let's see if there's anything I need to add here. The lag or latent phase is the time where there's no antibodies being produced, which takes anywhere from 3 to 6 days, like a week in the graph. And it includes all of these activities that I've mentioned: detecting the antibody, being activated by a T cell, the proliferation, the differentiation into plasma cells, and then the actual production of the antibodies. Covered all that already. Covered that. The secondary exposure, the secondary response is much faster. Thank you, memory lymphocytes. The response is much more robust. Not only is it quicker, but the antibodies are much higher levels. And this is also refers to active immunity, a direct encounter with the pathogen. Like I said, either by direct exposure or through vaccine. And then we have our memory cells. Passive immunity is where you get it from somebody else. So it could be natural or artificial. Natural, like as I mentioned before, the mother passing antibodies to the fetus through the placenta, or to the newborn by way of mother's milk. Artificial is where, that's what I was talking about, kidnapping my friend and maybe taking some of his blood and isolating the antibodies for my own purposes. This actually is a therapy. They're working on this. They started working on this a long time ago with the Ebola outbreak. Not even the most recent Ebola outbreak from about 4 years ago, but back in the late 90s, this was tried successfully, I might add. Let's see, serum containing antibodies against tetanus. So it depends on how the memory cells. Some of them last longer than others. I guess the ones like measles and such last your whole life. I had measles shot when I was a kid, never had one since. Others, like tetanus, don't last as long, and you have to have occasional booster shots. And I just recently had one earlier this month. And here's the example of snake venom. So an individual gets bitten by a snake. They use a variety of different ways. I don't know how they do it these days. They used to use chicken eggs to make the anti-venom antibodies against the snake venom. And so the person who's bitten, they would get the shot of the anti-venom, which would contain antibodies with binding sites shaped to fit that venom. Then let's see. It's temporary. When you receive the antibodies from somebody else, either artificially or naturally, you don't have the ability to make your own. So you don't have any memory cells. And that's the end of the immune chapter. Hopefully these videos took and I don't have to do it over again, because that was not fun. So then, for today and for the chapter on immunity, sayonara.